So hello once again. In this session, we are going to discuss about how different or various big data bioinformatics analysis methods will actually help you in understand the bioinformatics of infectious diseases. And we will also, in addition to discussing the application of different bioinformatics analysis methods, this program will also show you several examples of these analysis methods that are used in pathogen diagnostics, in evolutionary studies of the virus, and in the discovery of antiviral drugs and vaccines, and the design of antiviral vaccines, as well as to characterize emerging drug resistance across the different pathogens. So let's go over this briefly, go over the syllabus briefly. So by the end of the program, you will be able to explore relevant tools for characterizing sequence variation and sequence alignment at the whole genome level. You will be able to learn from peer-reviewed publications and example projects about how phylogenetic analysis helped in understanding the emergence and spread of pandemic like COVID and epidemic like Ebola viruses. So you will also be able to master key analysis methods and visualization tools that are related to sequence variation with functional properties of the protein. And these tools will help you understand the mutations that are critical for the pathogenicity and are critical for the functioning of the pathogens. You will also get an introduction to study the host's response by analyzing the transcriptomics data of the infected host compared to the healthy host. So we can also understand how actively the host is responding to the pathogen or to the infection in this case. So we will start again with an introduction to next-gen sequencing, and we will discuss about various NGS sequencing methods, and we will discuss about different data formats that are used in the sequencing, whole genome sequencing data, and we will discuss about exploring the Bioinfo server and exploring visualization of the reads that are mapped onto the whole genome sequence, in this case viral sequence or pathogen sequence, using visualization tools such as JBrowser and etc. Then we will follow this up with getting an introduction to multiple sequence alignment methods and phylogenetic tree analysis to understand the viral adaptations and the mutations that are characterizing the different pathogens. So we will also work with an example projects and examples of deadly mutations in Ebola and how that translates into different virulence or different effects of pathogenic effects of Ebola virus. So we will also get to compare multiple sequences at amino acid level, at nucleotide level, and also compare different methods for constructing phylogenetic tree analysis. We'll visualize the phylogenetic tree results using specific software that is available for tree analysis and also for understanding multiple sequence alignment methods. So in this hands-on session, this will be a follow-up hands-on session to the session that you studied before and discussed before. So where you will be shown how to build a pipeline so that you can perform multiple sequence alignment of viral genomes and a phylogenetic tree analysis using different phylogenetic tree methods. So you will also be given an introduction to different analysis pipelines, like selecting appropriate genomic sequences and selecting different pipeline methods that is suited for either FASTQ files or suited for FASTA files. So we will also analyze the output, discuss the pipeline results, and analyze how to perform the downstream analysis of the pipeline outputs and what to do if we have a FASTA file and FASTQ file for input. And we will also understand which databases to use to gather information regarding the viral sequences. And we will also see how to interpret the phylogenetic tree analysis that is the result of different phylogenetic tree methods, and how to understand the evolutionary relationships between the genomes in terms of evolutionary time, in terms of mutation, and in terms of birth and death of different strains that are yeah, that are with a varying degree of pathogenicity. And then from there, we will go to understand how viral transmission occurs cross species, and we will take this, or we will understand with the help of Ebola virus, and what are the specific insights we learned from studying the epidemic of Ebola, which is, as Elia mentioned, as one of the first epidemic that is studied, or that is real-time tracked using next generation sequencing data. And we will also understand the origins of these viral adaptations, and we will also extend this analysis to novel coronavirus genome sequence, so that we can compare the novel coronavirus SARS-2 viral sequence with the other related viral sequences. And we will get a good introduction to how the disease progresses once the virus infects the host. So what happens, and what are the functional domains of different proteins that means, for example, spike proteins and different proteins that are involved in the disease progression, and involved in helping the virus getting access into the cellular different cellular cytosol, and how this virus uses or hijacks the host cellular mechanisms to enable its own progression. And we will also understand how this how the disease how the pathogen evades the immune response of the host system in this session. And then what follows is a hands-on session of an example project, and this example project will compare the SARS-2 whole genome sequence with the other related viral sequence of SARS, MERS, and other to understand how related SARS-2 novel coronavirus whole genome sequence is with the other viral sequences. We will also take SARS-2 sequence from different hosts, so that we can understand the zoonotic origin of SARS-2 and how SARS-2 is related to, for example, to different parts of SARS-2, how it is related to different hosts, like from bat, from pangolin, etc. And then we will also understand how to analyze the mutation types, and how to analyze different mutation types like synonymous mutation, missense mutation, non-synonymous mutation, and how to calculate the rate of this mutation, and how to map these mutation rates onto the spike protein, for example, or onto the protein, so that we can get a visualization or hands-on visualization, or exploring the protein structures, and that is color coded, or that is mapped on for the mutation rate at its different position. So we will use use of Chimera and softwares like Chimera, Chimera X, and Pymol for this specific visualization. And then we will also understand what are the different low frequency mutation, and how will they actually enable the virus to maintain its virulence, and what are the different annotations on what are the different locations on the genome and proteome, different protein that contains these mutations, and how it relates to the accumulation of mutation only on the specific areas of, for example, spike protein and other related proteins, so that the virus adapts itself to maximize its virulence and remain fit in the evolutionary pressure. So finally, we will also understand the host side, or host response to such pathogenic infection. So we will get an introduction to transcriptomics data analysis. We will get an introduction to what is measured from the host side, and how we can perform a comparative RNA-seq pipeline analysis, so that we can identify key responses from the host side for the pathogenic interaction. So we can understand how different, for example, healthy hosts and infected hosts differ in terms of their immune response, and what are the different factors for considering vaccine design when after identifying a specific host's response pathways. So this program will also culminate in helping you to design a bioinformatics research and bioinformatics research project. You will be able to choose your topic for your project, and then again you will be explained where to extract data for example, related to infectious diseases and related to specific viral repositories. And then we will also get a lot of hands-on training on how to perform these analysis over the course of program that you can apply onto your own project. So I want to highlight 3 related publications that is published in 2022, and with the available number of data that is deposited in the public repositories for COVID. And these program and these analysis methods will enable you to learn, or will enable you to understand how these studies are performed, and then you can generate your own research question in the very relevant pandemic era, and which still helps you to understand different aspects of SARS-CoV-2 infection, and how big data bioinformatics is generally helping us tracking this pandemic, and also preparing us for any future attacks in the upcoming years. So we all know how the SARS-CoV-2 data, the rich data, has enabled understanding the different variants of concern or variants of interest, what are the different relationship between these variants, and how in real time, how these phylogenetics is being tracked, and how these different variants are related to each other. And we also can get an introduction to how the virus actually enables itself, or how this virus actually has an intra-host variation level that is not being explored in detail previously. So to get an understanding of on the scale of genomes that is being tracked with the COVID-19 response. So within a month of infection, we got almost an average of 1,300 genomes were being submitted to NCBI and being submitted to GISAID database per day regularly. And within the 6 months of start of the pandemic, GISAID had almost like 110,000 full-length SARS-CoV-2 genome sequences. So by the end of December, by the end of 2021, 2 years into the pandemic, we had 67,000 genomes that were deposited to the public viral genome repositories per day, daily. So currently, the repositories like GISAID, GenBank, NCBI, Virus, and COG UK, everything, all of these combined to contain over 6 million SARS-CoV-2 genomes in its entirety. So we can see where the phylogenetic studies and different surveillance studies are performed all over the world, and irrespective of different continents, all the continents contribute to tracking this viral pandemic in real time, and that is possible mainly through the analysis of big data analysis of the next generation sequencing data, and that has enabled us to understand and also keep exploring the data for future patterns and for further patterns that is so rich in its variety and throwing us a lot of surprises. So one of that is one of that surprise, or one of that key discovery is the in-host diversity of this virus, or its ability to adapt itself to in-host within the hosts based on the host response. So what this means? This means that the viral sequences that for example, that causes severe symptoms or whose virulence has an increased rate of virulence, has a different variation compared to the virus whose virulence has mild symptoms. So understanding this in-host diversity will pave way for us to arrest this development or this increase in the virulence factor of this virus, and thereby, even if the virus spreads from the host to the interhost or different to the different individual, we can actually make sure that not the most virulent virus is going to spread and going to cause infection for the other diseases. So we also get an overview in what is being currently studied and what is being clinically valued in the current pandemic scenario. For example, we value a lot on genomics and transcriptomics data only about the SARS-CoV-2. And from the virus's point of view, mostly the genomics data is what is being considered really informative. But we have a whole plethora of omics data from the host and also from the viral, from the pathogens responses, which can be integrated into a big multi-omic studies. And that aspect of this infectious disease scenario or pandemic scenario has not been explored in detail so far. So there are still a lot of opportunities there to develop methods that will effectively integrate, for example, the host's response to the pathogens and also the virulence levels of the pathogens to the host response, to study how these two partner in the effective transmission of the virus. So we have not even touched about how or what are the long-term effects of, for example, COVID infection. So into the third year of the pandemic, and we have still a lot of data that can be studied, for example, for the symptoms that prevail, what happens after 1 year or 2 years of 2 years of the infection. Already there are studies that that try to characterize the fatigue level, that try to characterize the loss of smell and taste level of the host individual after several weeks and several months of COVID infection. So this pandemic and also this infection has actually thrown up a lot of opportunities which are being currently explored at a different degree and at an increased rate. And this program will enable you to first understand and dissect all of these analysis methods, and then develop an analysis method for your own, for different data sets, so that you can understand the biology of these infectious diseases, or you can understand, apply these methods to another disease of your interest, and gain a lot of experience in the bioinformatics analysis of infectious diseases. So with that, I would